Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about shuttle vectors. Before that, will you please consider subscribing my YouTube channel? Your subscription is often a lot to me. First of all, let us know what is a vector. A vector is a DNA molecule, commonly a plasmid or a virus, which is used as a vehicle to carry foreign genetic material into a cell where it can be replicated and or expressed. Key types of vectors include plasmids, bacteriophages and artificial chromosomes. These vectors are used for cloning, protein production and also for gene therapy. Now the question is, what are shuttle vectors? A shuttle vector is usually a genetically engineered plasmid which can replicate in two different host species such as E. coli and yeast. These vectors are used for high capacity cloning and library construction. Because of the presence of dual replication of origins, and specific selectable markers DNA inserted into a shuttle vector can be manipulated and tested in two different cell types. A shuttle vector should contain at least two replication of origins and suitable marker genes for replication and selection in two different host organisms. Shuttle vectors include plasmids which can replicate in a prokaryote such as E. coli and a eukaryote such as yeast. One of the most common type of shuttle vector is yeast shuttle vector. Almost all of the commonly used Saccharomyces cerevisiae vectors are shuttle vectors which can propagate in E. coli and also in Saccharomyces cerevisiae. In addition, there are also adenovirus shuttle vector which replicate in E. coli and mammalian cells. As already said, shuttle vectors are used to transfer cloned genes between different organisms. These vectors are used for rapid cloning in the prokaryotic host, followed by its transfer to the eukaryote for functional analysis. One of the commonly used shuttle vector is yeast episomal plasmids, which are high copy number shuttle vectors. This figure illustrates the structure of YEP13, which is a yeast episomal plasmid containing two types of components such as yeast components and E. coli components. E. coli components, which are all derived from PBr322, includes two selectable marker genes that confer resistance to the antibiotics such as ampicillin and tetracycline. These two markers are used for selection in E. coli. The third component is an origin of replication where replication is initiated enabling the plasmid to replicate autonomously of the host chromosome. Yeast components that are present in YEP13 are ARS, Reg3 partitioning system, centromeric system, and leucine 2 gene. ARS means autonomously replicating sequence is the origin of replication in yeast such as Saccharomyces cerevisiae. It is a specific sequence at which replication of the plasmid is initiated enabling the vector to replicate independently. Reg3 partitioning system is another yeast component that ensures proper partitioning of the plasmids between daughter cells and also their stability. Third yeast component is centromeric sequence, which is used for proper functioning of the plasmid in yeast cells. Another yeast component is leucine 2 gene, which encodes the enzyme isopropyl malate dehydrogenase that catalyzes the third step in leucine biosynthesis. This gene is used as a marker gene for selection in yeast cells. All shuttle vectors also contains a multiple cloning site. It is a specific sequence which contains restriction sites for multiple enzymes so that the foreign DNA can be inserted at a suitable position. So friends, this is all about yeast episomal plasmid 13. This plasmid replicate in both yeast and E. coli. So by using the shuttle vector, the foreign genetic material or the transgene which is inserted into the multiple cloning site can be rapidly cloned in E. coli followed by its transfer to the yeast for functional analysis. Shuttle vectors have key uses such as gene expression studies, efficient molecular cloning, rapid amplification, and for studying complex systems such as DNA repair, mutagenesis, and recombination in mammalian cells. In addition, shuttle vectors are also used in yeast genetic engineering such as Saccharomyces cerevisiae research. So friends, this is all about shuttle vector. For more updates, please subscribe my YouTube channel, Sam Biology Class.